Theanine, also known as L-γ-glutamylethylamide and N5-ethyl-L-glutamine, is an amino acid analog of the proteinogenic amino acids L-glutamate and L-glutamine and is found primarily in particular plant and fungal species. It was discovered as a constituent of green tea in 1949. In 1950, it was isolated from gyokuro leaves. Theanine provides a unique brothy or savory, umami, flavor to green tea infusions. The name theanine without a prefix generally implies the enantiomer L-theanine, which is the form found in tea leaves and is a dietary supplement ingredient. Most studies have used L-theanine. The opposite enantiomer, D-theanine, has been studied less. The regulatory status of theanine varies by country. In Japan, L-theanine has been approved for use in all foods. Restrictions apply to infant foods. In the United States, the Food and Drug Administration, FDA, considers it to be generally recognized as safe, GRAS, and allows its sale as a dietary supplement. The German Federal Institute for Risk Assessment, an agency of their Federal Ministry of Food and Agriculture, objects to the addition of L-theanine to beverages. The European Food Safety Authority EFSA advised negatively on the use of L-theanine for improving cognitive function, alleviation of psychological stress, maintenance of normal sleep, and reduction of menstrual discomfort. Therefore, health claims for L-theanine are not recognized in the European Union. The chemical name N5-ethyl-L-glutamine and other synonyms, see box, for theanine reflect its chemical structure. The name theanine, without prefix, is generally understood to imply the L-S enantiomer, derived from the related proteinogenic L-amino acid glutamic acid. Theanine is an analog of this amino acid, and its primary amide, L-glutamine, also a proteinogenic amino acid. Theanine is a derivative of glutamine that is ethylated on the amide nitrogen, as the name N5-ethyl-L-glutamine describes, or alternatively, to the amide formed from ethylene and L-glutamic acid and its gamma-5 side chain carboxylic acid group, as the name L-glutamylethylamide describes. Relative to theanine, the opposite, D-R, enantiomer is largely absent from the literature, except implicitly. While natural extracts that are not harshly treated are presumed to contain only the biosynthetic L-enantiomeric form, mishandled isolates and racemic chemical preparations of theanines necessarily contain both theanine and its D-enantiomer, and from racemic syntheses, in equal proportion, and studies have suggested that the D-isomer may actually predominate in some commercial supplement preparations. Amino acid racemization in aqueous media is a well-established chemical process promoted by elevated temperature and non-neutral pH values, prolonged heating of camellia extracts, possible for oversteep teas and in undisclosed commercial preparative processes, has been reported to result in increasing racemization of theanine to give increasing proportions of the non-natural D-theanine, up to equal proportions of each enantiomer. Theanine is found primarily in plant and fungal species. It was discovered as a constituent of tea, Camellia sinensis, in 1949, and in 1950 a laboratory in Kyoto successfully isolated it from gyokuro leaf, which has high theanine content. Theanine is substantially present in black, green, and white teas from Camellia sinensis in quantities of about 1% of the dry weight. Deliberately shading tea plants from direct sunlight, as is done for matcha and gyokuro green tea, increases L-theanine content. The L-enantiomer is the form found in freshly prepared teas and some human dietary supplements. As a structural analog of glutamate and glutamine, the theanine in preparations, teas, pure supplements, etc., is absorbed in the small intestine after oral ingestion. Its hydrolysis to L-glutamate and ethylamine occur both in the intestine and liver, so theanine can be considered to function as a donor that supplies glutamate to the body. Glutamate can be metabolized to glutamine in astrocytes, a process catalyzed by glutamine synthetase, and can also be decarboxylated to GABA by glutamate decarboxylase, thus theanine can supply the neurotransmitter pools of amino acids. It can also cross the blood-brain barrier intact, and register pharmacological effects directly. Theanine is structurally similar to the excitatory neurotransmitter glutamate, and in accordance, binds to glutamate receptors, though with much lower affinity in comparison. Specifically, it binds to ionotropic glutamate receptors in the micromolar range, including the AMPA and kainate receptors and, to a lesser extent, the NMDA receptor. It acts as an antagonist of the former two sites and is an agonist of the latter site. Theanine also binds to group I mGluTars. In addition, it inhibits glutamine transporters and glutamate transporters, and thus blocks the reuptake of glutamine and glutamate. Lastly, theanine elicits umami taste, and this effect has been found to be a consequence of the fact that it directly binds to and activates the T1R1 plus T1R3 heterodimer or umami, savory, taste receptor. Theanine increases serotonin, dopamine, GABA, and glycine levels in various areas of the brain, as well as BDNF and NGF levels in certain brain areas. However, its effect on serotonin is still a matter of debate in the scientific community, with studies showing increases and decreases in brain serotonin levels using similar experimental protocols. It has also been found that injecting spontaneously hypertensive mice with theanine significantly lowered levels of 5-hydroxyindoles in the brain. Researchers also speculate that it may inhibit glutamate excitotoxicity. Able to cross the blood-brain barrier, theanine has reported psychoactive properties. Theanine has been studied for its potential ability to reduce mental and physical stress, improve cognition, and boost mood and cognitive performance in a synergistic manner with caffeine. Theanine has also been shown to inhibit peroxidation in vitro and help protect against oxidative damage induced by chronic restrained stress. A Natural Standard monograph that reviews current research on theanine reports that it is likely safe in doses of 200 to 250 mg up to a maximum daily dose of 1,200 mg. Though some people use theanine for these purposes, Natural Standard rates the evidence to support the usage for anxiety reduction, blood pressure control, and mood improvement as unclear or conflicting scientific evidence, and the evidence for improved cognition as fair negative scientific evidence. Many of the studies of theanine were done in combination with caffeine as found in tea. While the studies found that the combination had some effect on mood, the studies found that theanine alone had little effect. A review by other researchers of a small set of trials concluded that there are benefits of L-theanine in reducing acute stress and anxiety in people with stressful conditions. In 2001, the German Federal Institute for Risk Assessment, Bundesinstitut für Risikobewertung, BfR, objected to the addition of isolated theanine to beverages. The institute stated the amount of theanine consumed by regular drinkers of tea or coffee is virtually impossible to determine. While it was estimated the quantity of green tea consumed by the average Japanese tea drinker per day contains about 20 milligrams of the substance, there are no studies measuring the amount of theanine being extracted by typical preparation methods, or the percentage lost by discarding the first infusion. Therefore, with the Japanese being exposed to possibly much less than 20 milligrams per day, and Europeans presumably even less, it was the opinion of the BfR that pharmacological reactions to drinks typically containing 50 milligrams of theanine per 500 milliliters could not be excluded. Reactions such as impairment of psychomotor skills and amplification of the sedating effects of alcohol and hypnotics. In 2006, a study found no consistent, statistically significant treatment-related adverse effects on behavior, morbidity, mortality, body weight, food consumption, and efficiency, clinical chemistry, hematology, or urinalysis in rats fed high doses of theanine for 13 weeks. Large studies in humans have not been undertaken. However, several smaller-scale studies, fewer than 100 participants, have shown increased alpha wave generation and lowered anxiety, along with benefits to sleep quality in people with ADHD. The combination of theanine and caffeine has been shown to promote faster simple reaction time, faster numeric working memory reaction time, and improved sentence verification accuracy. Theanine has been reported to raise levels of brain serotonin, dopamine, and GABA, with possible improvement in specific memory and learning tasks. A study of tea bags sold in British supermarkets in 2011 found that the tea bags containing the most L-theanine per cup, 24 milligrams versus 8 milligrams per cup, were the lower quality brands containing black tea, with a supermarket brand of black tea having the highest theanine content. The study demonstrates that brewing time is a major determinant of the amount of L-theanine extracted. Addition of sugar and small quantities of milk make no significant difference, while larger quantities of milk reduce the measured theanine content.